Alrighty, we are officially five past, so I think it's fair that we go ahead and get started. I'm sure folks will still be trickling in, but first I wanted to thank everyone for taking time out of your busy weeks to join us today, and we're really excited to talk about 5 years of Black and Neuro this week and all of the work that we've done, and what a great way to kick this off by having Dr. Rasheen Powell talk about his work. Um, before we begin today, I'd like to acknowledge that I am joining you from territory of the Muscogee Nation. I invite everyone to take a moment to reflect on the lands that you are joining from and your relationship to those lands and the indigenous people who have and continue to inhabit them and were forcibly removed. Even as folks of the African diaspora, we're still required to think about our place in injustice in the world. The next thing I'd like to mention is that we do have automatic closed captioning, and it's enabled in the Zoom call, so you can choose to view or hide closed captioning by clicking the live transcript or show captions button. Should be labeled CC. And if you have any issues accessing these captions, please message the ghost moderator in the chat. All right, I do have the sniffles today, so it'll be the sneeze heard around the world. Um, so the next thing we wanted to put on your radar is that we'd love to invite everyone to join us for Black and Neuro Week 2025, happening at the University of DC from September 10th through 12th. And we're super excited for all of the work that's coming together and all of the efforts that our team has put together to bring our first in-person conference. And programming will be available virtually and in person. So there's plenty of opportunity for you guys to learn and connect with one another. And it's definitely going to be dopamine, as I like to say. And a registration link will be posted in the chat for you guys. So if you have any questions for Dr. Powell today, you can post them in the chat as well. And when once we have time at the end, then we'll allow you to unmute yourself and you can ask your questions to Dr. Powell. So I'm really excited to introduce our speaker today. So we have Dr. Rasheen Powell, a postdoctoral fellow at Harvard University. Always curious, Rasheen has been interested in science since his elementary school days. He has maintained this curiosity throughout his life, starting with summer research camps in the city of Buffalo. And this curiosity was furthered by other extracurricular endeavors at the University of Buffalo Medical Science Technology entry program, as well as a four-year high school research program at the Hop, oh, sorry, I hope I don't say this wrong, Hauptmann Woodward Research Institute. From learning how proteins work to learning emergency medical techniques, which is so incredibly important, Rasheen was exposed to boundless potential opportunities for biomedical science at an early age, and this sparked his curiosity. So, however, he didn't always want to join the field of neuroscience, surprisingly. So, he had never taken a proper neuroscience course until graduate school, and his undergraduate studies were in pharmacology and chemistry. Nonetheless, he immediately pursued a PhD at the University of Buffalo, where he excelled in his studies and received a PhD in pharmacology under the mentorship of Dr. Aaron Bhattacharjee in 2021. His work was centered around investigating novel targets for treating chronic inflammatory pain. From there, he moved to Boston and immediately began a postdoc in the lab of Clifford Wolf, where he continues to expand on his PhD thesis work investigating novel opioid molecules for treating neuropathic pain. So without further ado, he'll be giving a talk titled Pain, It Hurts, and It Helps. So please join me in welcoming Dr. Rasheen Powell.
Awesome. Thank you. Thank you for that introduction, Victoria. Very nice. Very, very flattering. Well, I was really kind of mulling over what I wanted to title this talk because I myself am still technically a early stage investigator. However, I do feel as though I've lived a little bit of life and that knowledge, that whether it be academic or life values, I think would be beneficial just to disseminate out to the Black and Neuro community. So today I'm going to take a little bit of talk a little bit about my work around pain, the aspect of how it hurts and how we are trying to develop new ways to treat it, but also the pain of actually being a scientist and the pain associated specifically with being Black in neuroscience and how that pain can hurt at times, but it can also be a driving force for pushing you to that next stage. So there we go. What is pain, right? A lot of these questions may seem to be kind of nebulous or they may seem to be a little philosophical, but the nexus point of what I do in the lab and outside of the lab is really mulling over this question, what is pain? And today I'm just going to present to you, like I said, a story about life and also a story about neurobiology. So who am I? I'm just a guy who does science. I was born in Savannah, Georgia. I was raised in Buffalo, New York, and I went to school at the University of Buffalo. I got a dual degree in the pharmacology and toxicology program, as well as a BA in chemistry. And as was said in the intro, I went straight into a PhD program, all gas, no brakes, dove headfirst into it. And then after the cessation of that, and I got my PhD, I immediately moved to Boston and started a postdoc at Boston Children's Hospital and joint um, appointment with Harvard Medical School in the lab of Clifford Wolf. Now, as you can see, I really just kind of fast tracked a lot of this. I wish I took a little bit of time in between, but there are some reasons as to why I really want to go gung ho into science. I always love science. Science has always been something that I've cherished and something that I always find very interesting. The curiosity of being able to ask a question and then go through the steps of answering that question, a question that many people may have had or no one's ever had before, and then becoming the person, in this sense, the individual who becomes that specialist in that highly specific question. I always thought that was very, very encouraging and very astounding for me. So I really did keep with the sciences all throughout my life. So what did I do? Well, as Victoria said in the introduction, I am a pain researcher. I'm looking and trying to have a mechanistic understanding of how ion channels specifically are moving in neurons during pain. And my research in my PhD was focused on chronic inflammatory pain. My research now is in chronic neuropathic pain. And the ultimate goal is to develop non-opioid analgesics to disrupt interactions between the ion channels and these accessory proteins that may bind to them and promote their movement to and from the membrane. But one thing that I did learn, and one thing that I'm sure a lot of people know, is that pain is not just in a lab setting, right? Pain is not something that you can distill down into its quantitative and qualitative measures with an animal model or with a protein-protein interaction. Pain can actually come in many different forms, and often it does. And the most common types of pain that we may come across may be broken down into three main aspects, right? You have your emotional aspect, the mental aspect, and the physical aspect. And this isn't NFL, NBA, or this isn't anything. You're not standing on the world stage in a sports competition. So when you go into science, you're really not going to get that much physical pain. But I will say that the emotional and the mental pain can be very overwhelming at times. And I'm just going to go into some examples as to what do I mean by that. And as I said, like science can really exacerbate and really amplify a lot of these different types of pain. So the emotional pain, right? These are going to be questions or some things that I actually dealt with when I started my scientific career, specifically my PhD. My undergraduate, it was studying, answering questions on tests and quizzes and stuff like that. But once you start to take science as a career and you go into a postgraduate space, it does get a little difficult. And some of the internal struggles that I dealt with, or the emotional struggles I dealt with, were, do I really want to do this? Do I want to be a scientist? Do I want to sit in a lab? And do I want to be there late, get there early, not have weekends, miss out on friends, family outings, and things like that? Another aspect is the feeling of isolation. I myself, my parents are not scientists by any stretch of the imagination. So when I tell them what I do, they encourage me, but they don't really know what I do. They think that I play with mice, that it's just me in a room and I'm just kind of having a grand old time with these little fur balls and I come home and that's it. But you really do get this feeling of not being able to explain what you do, not being able to explain the struggles associated with what you do with friends or family. So you kind of get this feeling of isolation, this idea of, well, am I alone in this? And then another aspect that came in a little later in my scientific career during my PhD is this imposter syndrome, right? Am I good enough? Is this something, am I at the stage where I need to be to move on to the next stage, right? Or am I comparatively compared to other individuals, am I keeping up? And these are some of the questions that I had asked myself. Where I am today, you can probably tell that I got over it, but in the moment, it does feel very difficult, very difficult. And I will say that some of the things that get you through the emotional aspects of this pain is, just talking. It could be, even though that your family or your friends may not understand what you do, you can phrase it in a sense or in a way that allows them to uh, get what you're saying, allows them to come up to and meet you halfway so that you can explain why it's difficult, not the nitty gritty details, but really they can empathize with you in that aspect or even sympathize with you in that aspect and offer, just a ear to listen. Because sometimes you just need to get things off your chest. Now, the mental pain, this is something I think is a silent aspect of the scientific space that is uh, not appreciated enough. You know, Emotionally, yes, you're supposed to be a scientist who is very uh, quantitative. You're really looking at empirical data. There's no room for emotions. But mentally, as a scientist, that's you can't keep that up 1,000% of the time, right? So this idea of burnout, burning the candle at both ends, this idea of can't I just quit? It'd be much easier. I can, drop out of this program. I can do something that's a lot easier and, live that way. But if you really want to be a scientist or if you want to be a neuroscientist, you can't just quit, right? I mean, you can if you really want to, but if that's something that you really want to do, something that you've vested time, effort into, maybe what you're researching isn't appropriate for your mental state. Maybe that's not a field that you want to dive into. Maybe there's something else in neuroscience that you're interested in, or maybe the lab environment isn't the best. Your PI may be demanding things that you just can't produce because we're only human. There's only 24 hours in a day, and not everyone can wake up, eat, drink, sleep science. You need to have those periods where you can do your science and then you don't do science. What you do inside of lab is equally as important as what you do outside of lab. So this idea of burnout, I brushed up against it very closely during my PhD, but my PI was very cognizant. He said, listen, you've been working really hard. Why don't you take a couple of days off and then come back and we can tackle this problem on the back end? And just hearing him say that really did, I think, save the mental aspect of myself as I was discovering who I want to become as a scientist. That idea of social comparison, why can someone else that I know in another lab publish so fast or get this great data and I'm over here floundering and flopping around and not generating anything? That idea that I need to be able to keep up with the Joneses or keep up with this person in that lab or so and so published this recently in this high esteemed journal. Any none of that really matters. Not all PhDs are created equal. You move at different paces, and that's perfectly fine. The science is the science, and it works as fast or as slow as it needs to go. And then lastly, and the most important, I think the strongest aspect is science is really unforgiving. When you're doing something new or you're investigating a new field, you're asking questions that some very little or no one has ever asked before. So you will fail. It's not a question of if you will, it's a question of when you will and how frequently you will fail. But as a scientist and you're developing these your skills and you're going through this mental anguish, you have to realize that everyone goes through this. There is no scientist who is 100% perfect, never failed anything. I fail Western blots. I'm an electrophysiologist, so I fail passion quantum sales all the time. I mean, recently I've failed a couple of electrophysiology experiments, but in the beginning, it can be very disheartening and can really draw and drag down your mental space and really be a negative. But you have to understand that this is a it too shall pass. Essentially, it's not something that will follow you throughout your career. Just because you failed one thing, that does not then translate to your aptitude as a scientist. It actually just is a testament to how difficult science can be. So you don't want to take these emotional and mental shortcomings or these emotional or mental roadblocks and don't apply them to your entirety or your career as a scientist. They're very transient and they will pass. So, speaking on the pain aspect. Here are some quotes that were given to me throughout my scientific career that really pained me, right? Here's one: you only got accepted because, kind of alluding to me being black and, traditionally underrepresented in sciences. I don't know why you're still here. You already got what you needed from me. This was told to me by a PI that I was doing undergraduate research with in organic chemistry. Our, uh, we didn't mesh well as far as our, let's say our personalities. We'll just leave it at that. But she had two labs, and I was working in the auxiliary lab, but she never really saw me, so she was under the assumption that I was just being lazy and not coming in. But she didn't know that I would come in at night when I had a gap between my last class and before I had to go to work. I worked night shift at a horse ride or horse, excuse me, an off-track betting place where you place bets on horses. So I had very small hour time windows of about 3 to 4 hours where I would come in the evening, do some work, and then go to work, and then come back and go back and forth that way. But she thought I was just kind of there for the letter of recommendation. So she kind of snapped at me one day, and I was like, you know what? This isn't for me. Thank you for the opportunity, but I have to go. Here's another one. I don't think you would thrive in my lab. You did your undergraduate research in chemistry. I do neuroscience. So this was told to me by a PI while I was looking for a lab to rotate in. We had a program that would pay for us for the first 2 years, and they offered us the opportunity to rotate throughout different labs. And I had approached this PI and said, hey, um, I'm very interested in what you do. I was wondering if I can just do a rotation. And she said, no, for this reason. And I said, okay, sure, no problem. And then come to find out, a student she allowed rotate in her lab was who had a PhD in or excuse me, had a master's degree in chemistry. Did his first rotation in her lab. So at that point, I was like, ooh, that one kind of hurt. It stung a little bit because, what does he have that I don't have? And then you start getting to that mental game of, am I good enough, or am I able to keep up with other people and stuff like that. And then the last one that I want to leave here is that you just have to keep your head down and keep doing good science. Everything else will follow. So this was actually told to me by my PI. He's a great guy. This was during the George Floyd protests around COVID and everything, and we were having discussions on what it means to be Black and a scientist at the same time, and how even though in the lab I'm seen as a scientist, once I take that lab coat off, everyone else in the world, I'm just another Black man on the street. So he may know that I'm a scientist, but they just see me as whatever they stereotype a Black man to be. So this one hurt me for a different reason, not because it was a negative slight towards me, but because it felt like my identity was being minimized and reduced down to just a scientist. And in a scientific space, it may be appropriate, but for my lived experience, that's just not who I am. A scientist is only a small part of who I am as an individual. First and foremost, I'm Black. I was born in this world Black. I will die Black. A scientist is just a label that I'll carry on for a portion of my life. But that's all just to say it didn't really matter because, and these are the people that who said these things. But at the end of the day, I finished my PhD with a excellence in thesis dissertation award, so the Dennis Higgins Award. So I was recognized as an excellent dissertation for the pharmacology and toxicology department, and I also received the distinction of having an excellent thesis across all biomedical sciences dissertation submitted that year. So even though there were a lot of things that pained me throughout that entire process of getting my PhD, at the end of the day, it was me on the stage holding up the awards and smiling. So this is just to say that it's going to be difficult. This is to say that the pain will be there, but how you react to that pain and how you take that pain and convert it into something that may be a learning experience or something that can propel you forward is invaluable. So don't take it as in the moment, it's going to hurt, but just know that that hurt, it does help on the back end. So uh, that's my intro. That's my spiel about my kind of the pain, hurting, and helping aspect. But now let's talk about this from more of a neurobiological sense. So I'm going to switch gears and talk about the neuroscience a little bit and how my experiences and my background actually helped me to kind of recontextualize everything that I know about pain and treating pain. What we know about pain as far as a sensory detection mechanism is that you have some type of stimulus. That stimulus may be painful, and then you have to remove yourself from that stimulus. So think back to when you're a kid, and you may have grabbed a hot pan, and then you realize, oh, that's hot. You dropped it automatically, and then you shake your hand a little bit. It burns, it hurts, you may cry, and then you realize I'll never do that again. And that's what pain is. Pain is a mechanism to teach us what to do, what not to do. It's an alarm system. It helps us survive. But the problem is that some individuals are in a state where that pain becomes chronic or it's inappropriate for the stimulus that's being presented. And then that's where we start having issues. So this is an image that was actually in the Treatise of Man by Degas that was published back in the 1600s, where he had this idea that pain is just a rope that kind of rings a bell in the head. And, it's actually not too far off the mark, but we know it's not just a series of ropes and pulleys that are pulling a bell in your brain. It's actually a lot more complicated than that. We have the brain, the spinal cord, peripheral neurons that are responsible for responding to different stimuli. So, although he was kind of on the money, he was a little ways off. And the complexity of this quote unquote pain pathway is actually kind of driving the difficulty in treating it appropriately. So the pain type that I'm looking at now is specifically neuropathic pain, where I'm looking at lesions or disease of the somatosensory system through direct injury, infection, chemical exposure, compression entrapments, or stroke, and various other insults to the nervous system. This neuropathic pain causes debilitating changes in the quality of life. Um, Patients often complain of spontaneous pain, evoked pain, pain that wakes them up out of sleep, loss of quality of life, and inability to work. And may have some mental deficits like loss of memory, loss of ability to make decisions. So neuropathic pain is just not ooh, that hurts and it's continuously hurting. It truly infiltrates every aspect of a patient's life and decreases their overall quality of life. When it comes to treating pain as it stands now, we have a few options. Some of them work. The issue is there's not one that works 100% of the time. So some of them work some of the time, but none of them work all the time. The first line or classical first line therapeutic options are your gabapentinoids like gabapentin or pregabalin. Your second line options will be opioids, local treatments with capsaicin or lidocaine, and your third line options are actually going to be more opioids. So you'll notice when it comes to treating neuropathic pain, opioids are not the first option. As we know, or we may have heard about the opioid crisis in the U.S., uh, although engineered, it is still a something that we're dealing with the fallout now. Opioids are the traditional go-to for a lot of types of pain, but our options are currently limited, and we're kind of just going back to the well and hoping these opioids work. But for about 70% of patients, opioids have no efficacy. So my job as a pain scientist is to come up with something new, something inventive. And the way that I do that, sorry, is reevaluating where's the pain coming from and how can we treat that. So the number one or the molecular driver of pain in nociceptors or these pain sensing neurons are going to be your voltage gated sodium channels. So you're interested in neuroscience, these voltage gated sodium channels are going to be at the kind of the apex of everything you know about neuroscience as it pertains to neuronal activity. They are consistent of 10 members that are composed of an alpha subunit class and a beta subunit class. The alpha subunits form the pore or where the actual ions traverse. The beta subunits are auxiliary and interact with the pore, forming subunit in various capacities. And each subunit, alpha subunit, has different gating kinetics that partitions it to a specific function in the action potential. So here is just a schematic mock-up of what these different sodium channels do and what their overall goal is in the action potential generation. You can see NAV 1.2, 1.3, 1.6, 1.9, 1.1. Those are subthreshold. NAV 1.7, which is setting the threshold at which the action potential will be fired. And then you have NAV 1.8, which is responsible for the overshoot or the upstroke of the action potential. What's really great or what's really interesting is that NaV 1.8 is the predominant contributor to the action potential in nociceptors specifically. So NaV 1.7, 1.9, and 1.8, the preferential expression in those pain sensing neurons. So these have been very attractive when it comes to treating pain. But when we look at where they are in the neuron, they actually show different levels of expression in different compartments. So it's not only that these three ion channels are found most heavily in pain sensing neurons, where they are within the pain sensing neuron is completely different depending on which isoform you're looking at. So NaV 1.8 is going to be in the periphery, in those innervating tissues. The NaV 1.7 is going to be in the presynaptic terminals leading into the spinal cord. And NaV 1.9 kind of has like this spotty distribution across different areas, mostly in the periphery and a little bit around the cell body, but mostly in the periphery. And this kind of begs the question, well, why do they have this distribution? And does this kind of lend to a maybe tap something we can tap into as pain scientists to develop something new, novel that targets this and may have some type of impact on pain. Well, there have been as far as an industrial effort, there's been a lot of money spent on this for 20 plus years, and they all had the same idea. Well, if we can block these channels, then we can stop pain. And recently, if you're into this space, you know that Vertex Pharmaceuticals just had their NaV1.8 inhibitor approved for acute pain, Dronavix or suzetrigine. That is a NaV1.8 blocker. It blocks the pore, and or excuse me, it allosterically modulates the activation of the channel and prevents sodium flux. And they showed in their clinical trials that it does significantly reduce pain in pain patients who've undergone surgery. The problem is that although it decreased it, for me, it didn't decrease it enough. We know that NaV1.8 is a very strong driver of pain, but their data is not really convincing. So now that begs the question, is there another way we can target these target these NaV channels while getting a therapeutic that is non-opioid in nature, but still targeting the same channel? So in my neck of the woods, I like to look at proteins that bind to the NaV channels, not the NaV channel itself. We know a lot about those NaV channels, but we don't know a lot about the auxiliary or the accessory proteins that are binding to them and affecting how they gate, and more importantly, how they actually get to the membrane. So one family of these proteins that I'm looking at are known as the ankrins. These are these large scaffolding proteins that bind to a bunch of different things, but most importantly, they can bind to NaV channels, and they are known to bind to NaV channels in great degrees. In the classical neuron, you have your node of Ron VA, your axon initial segment, and that's actually where you find these anchorins. They're found in these very discrete subcellular localizations. And right there with the anchorins, you'll have NaV channels, or these voltage gated sodium channels. In the central nervous system, you'll find NaV 1.6. In these areas, however, in other areas of the body, like in the heart, you'll find NaV 1.5 binding to ankrins. You'll find NaV 1.6 binding to ankrins and all across the brain. And mutations in this interaction actually does lead to decreased electrogenesis in these electrically excitable cells. So in the case of the heart, cardiomyocytes actually lose the ability to have that cardiac action potential properly propagate along the membrane. And in the neurons, they actually have disruption in the propagation of action potentials. So it shows that the association of these anchorins with these NaV channels is extremely important for the function of the cell type as well as the overall tissue. So then that begs the question, well, can we disrupt this interaction between NaV 1.8 and the anchorins and get a reduction in pain? So there's a couple questions. The first one do sensory neurons have anchorins and have NaV1.8? Yes, they do. Do they have the discrete subcellular localizations? That one's a little more tricky because in the DRG, where the sensory neurons are housed, the dorsal ganglion, there are some that have myelin that have the node of Ron VA and the axon initial segment, but then you have some that don't have nodes of Ron VA. And they have the axon initial segment, but can they still engage in this mechanism of ankyrin binding to the NaV channel and allow for proper electrogenesis? Well, that is the big question. So the hypothesis that I initially had when I started this project was that these nociceptors are leveraging a unique interaction between these two proteins for nociception. The questions were, is NaV1.8 and anchored interaction necessary for proper electrogenesis? Does this interaction underlie nociception in neuropathic pain? So let's start answering these questions. Well, the first thing you got to do is, are these proteins being expressed? So in my system, I use dorsal ganglion neurons that are cultured from mouse embryos, and I can culture them, mature them in a dish, and then I can do a bunch of different experiments. In this one, I'm just doing a little bit of immunocytochemistry to see where these proteins are. And the first two that I looked at were ankyrin B and ankyrin G. These two ankyrin isoforms are highly expressed in the nervous system. There's another ankyrin R. I haven't found a lot of data showing its expression, and in my hands, I have yet to see expression at this level in my culture system. So I don't really regard it when I'm doing my future experiments, but it is there. The one thing we can see is that ankyrin B and ankyrin G are found in these neurons in very high amounts, and they're exactly where you expect them to be. When we do co-localization staining, I'm looking at ankyrin B in cyan and NaV 1.8. the ion channel of interest in magenta. And then on the right, we have ankyrin G in yellow and NaV 1.8 in magenta. And we can see wherever an ankyrin or wherever the NaV channel is being expressed, those ankyrin are right behind it, which is great because it suggests that there is that interaction between the ankyrin of different isoforms and NaV 1.8. So since I know that these proteins are being expressed in my culture system, now it's time to test to see if this interaction is necessary for proper electrogenesis in the nociceptors. And to kind of, cheat a little bit, they are technically, but they're not really. So the way that I did this was using what's known as a lipidated peptide. It's a peptide about 14 amino acids long that has a hydrocarbon covalently linked to the N-terminal. This allows that peptide to be embedded into the cell membrane of the cell, and it mimics the sequence that binds to ankyrin. It mimics the NaV 1.8 sequence. So it directly competes with NaV 1.8 for ankyrin, and since it's in such high amounts, it'll outcompete NaV 1.8. It will bind to ankyrin instead of NaV 1.8, and then I can see what is the change or what is the output in terms of electrogenesis in these cells. So I'll have three groups in this on this slide. One is the wild type, one is the scrambled peptide, and one is the NaV 1.8 mimicking peptide. And here is just showing the total sodium current through these cells at different membrane voltages. And we can see that when I give this peptide to the cells, there's actually no difference between any of the groups. So the wild type is just like the scrambled, which is just like the NaV 1.8 peptide. When I look at the peak sodium current, so the maximal amount of sodium going at negative 20 millivolts, we can see that it's exactly the same. And then when I look at the actual traces, you see that there's really no difference between any of the groups. So it would suggest that this peptide is actually doing absolutely nothing, and the interaction between NaV 1.8 and ankyrin is non-consequential. When I add TTX, which is a tetrodotoxin, which is found in pufferfish, it actually binds and blocks specific subsets of those NaV channels. So there are 10 different subunits, but 3 out of those 10 alpha subunits are resistant to TTX. So NaV 1.8, NaV 1.9, and NaV 1.5. NaV 1.5 is found in the heart, so it won't be in high amounts in my culture system. However, NaV 1.8 and NaV 1.9 are found in these cultured neurons, so they'll be in high enough amounts where I can do a similar sodium channel, or excuse me, sodium current determination. And when I isolate just those currents, just the NaV 1.8, NaV 1.9 current, I see no change. And again, when we look at the peak current as well as the traces, there's really no difference. So this had me thinking, it's like, okay, well, there's two possibilities. Either the interaction between NaV 1.8 and ankyrin isn't needed for anything, or the peptide just doesn't work. So as a scientist, you have to vet all possible opportunity or all possible contingencies. And the first thing that I did was, well, take a look to see if I were to take these neurons and then challenge them with a molecule like a stimulus to make them excitable, can I see a difference? So, and this is just a schematic showing that the ankyrin binding to NaV 1.8, when I have the peptide on, it doesn't really do anything. But uh, one thing that's really cool about these neurons is that they don't exist as a single state. They're actually flowing through multiple different states. They're highly plastic, if you ever heard of neuroplasticity. And nociceptors have the ability to switch from being very quiet to firing a lot of action potentials if the conditions are appropriate. So one thing we can do as the manipulation in the lab is expose these neurons and incubate them with a compound known as 8-bromocyclic AMP, which mimics the cyclic AMP necessary for PKA activation. And once you have PKA activation, protein kinase A, it'll phosphorylate a bunch of different proteins and actually bias the cell to a hyperexcitable state where it's no longer quiet and is firing multiple action potentials. And that frequency of firing is our biological correlate to pain in the lab. And what we can see is that cells that get the 8-bromocyclic AMP actually show an increase in the total sodium current. They show a decrease in Rio base, or the total current necessary to elicit an action potential, and they just fire more action potential. So they are indeed signaling in a pain-like state, which is great. So what I did was I took this compound and then I just redid the previous set of experiments. And what we can see now is that actually, surprisingly, the cells that receive the NaV 1.8 peptide show a significant decrease in the total amount of sodium current. They show a decrease in the peak sodium current, and you can actually see that in the traces. And when I add that TTX to block all those TTX sensitive sodium currents, we can see that that current reduction persists. So that suggests that the reduction in current, the TTX sensitive. And then that is also recapitulated with the peak analysis and also the traces. So it's great that the peptide works and it's reducing the amount of sodium currents when PKA is activated. But is there actually a functional change in those nociceptor firing or those nociceptor properties? So here I'm just looking at current clamp, where I'm looking at different types of things associated with the action potential firing of the cells. So like the number of action potentials and also the real base or the minimum current necessary to elicit an action potential. And as was seen previously, without any PKA, there's absolutely no difference in any of the groups. When you look at the actual action potential, they look very similar. There's a slight reduction in the height of the action potential, but things like um, the threshold potential, the action potential half width, there's really no difference. So for all intents and purposes, the peptide does nothing to these cells alone. However, if I again pre-incubate with 8-bromocyclic AMP to make these cells hyperexcitable, we can then see the difference. Now the cells that receive the peptide are less excitable. You see that they require more current to fire the first action potential, and you can start to see large changes in the action potential waveform, where you have a reduction in action potential height, in a reduction in the actual width of the action potential, showing that it's much thinner, suggesting a reduction in that NAV 1.8. So for all intents and purposes, when you have ongoing PK activity, this peptide is able to induce a reduction in NAV 1.8 metrics. And here is some immunofluorescence data showing the specificity of this peptide. So here are two halo tagged NAV constructs. One is NAV 1.7, which is TTX sensitive, and one is NAV 1.8. And what happens when I have cells that are expressing this NAV 1.7 halo tag, and the cells that are expressing the NAV 1.8 halo tag, I can look at these cells over time and expose them to 8-bromocyclic AMP and see how these channels move in the membrane. And what's really cool is that both of these cells receive the NAV 1.8 peptide. The NAV 1.7 construct, you can see it's very consistent from 0 to 10 minutes. The NAV 1.8, you actually see there's an increase in the NAV 1.8 signal, which is really cool, suggesting that trafficking event. But as time goes on, at minute 20, you can see that the NAV 1.7 stays exactly the same, but the NAV 1.8 goes away. And then that persists for 30 minutes. So this peptide is actually able to uh, change how the channels are moving in and out of the membrane. So what I think is happening is that when you have PKA active, the NAV 1.8 NAV 1.8 channel is being removed or endocytosed because ankyrin is no longer binding to it and protecting it from endocytosis. Instead, it's binding to the peptide. So that's all well and good. I can get this to work in a dish, but can I get it to work in animals? So here is some examples of this peptide actually working in animals. This is a model of acute inflammatory pain. You inject a molecule known as zymazin, which causes an immune response, and the animal's paw will swell up. So these are animals that receive either the scrambled peptide or the NAV 1.8 peptide. And we can see that the animals that receive the NAV 1.8 peptide are showing a increase in their thermal withdrawal latency, so how long they keep their paw on a thermal stimulus, statistically different. And we can see that there's also a difference in their mechanical sensitivity 24 hours after they receive the zymazin. So it corresponds to the thermal data, which is really cool. But what I think is the coolest thing is that when you look at how quickly the animals recover over time, there's actually no significant difference between the scrambled peptide and the NAV 1.8 peptide, showing that the peptide is not changing how the inflammation is progressing. It's solely changing how these animals, how their neurons are actually signaling for pain. And then if we look at a model of neuropathic pain, this is a model of neuroma where you can take the sciatic nerve of a mouse and you can cut it and completely transect it. You'll get this bulb of disorganized axons that'll spontaneously fire and signal and send these signals to the brain and signal for pain. And we can see that in animals that undergo the surgery on the ipsilateral side, they show this very progressive response to mechanical stimuli, whereas on the contralateral side, there's no response whatsoever. So you can get a unilateral response in these animals and just kind of stimulate that one side of the animal, see how they respond, and then give them a drug to see does that drug impact the response on the ipsilateral side? So what we did, and with the help with another postdoc in the lab, is I took my drug and I we put it in a little cuff that goes around the neuroma, and then we tested the behavior of those animals to see, does this drug have any effect? And what's really cool is that the animals that receive the peptide show a progressive decrease in their pain responsiveness that then prolongs and stays prolonged for about 9 days before it naturally recovers. And we had a day where it was really exciting, where one animal actually went to zero. So it went from responding 100% of the time. You poke or you give the stimulus 10 times, and then uh, you're doing the percentage of how many times they respond. And then there was one that went from responding 100% of the time to responding to 0% of the time, which was amazing. So that was really exciting day. And then we did a little bit more characterization where we're looking at the actual calcium transients in live animals using these VGLUT6 GCAMP6 mice, where we can see what neurons are firing when we stimulate the neuroma. And we can see that animals that got the peptide, the NaV1.8 peptide, show the number of cells that are responding decrease significantly, which is great. And also the number of spontaneous bouts of behavior that we were observing were significantly decreased in animals that received the NaV1.8 peptide. So altogether, the peptide is exerting an analgesic-like effect in both that model of inflammatory pain and this model of neuropathic pain. And then when we actually look at the neuroma and look at the staining, here I have peripherin, which is going to label the nociceptors, or those cells that signal for pain. And in red is ankyrin 3 or ankyrin G. And you can see that in these areas, you have these small puncta of ankyrin 3, suggesting that, oh, there may be these clusterings of NaV1.8 and ankyrin in the disorganized neuroma, suggesting that mechanistically, the after injury, and you'll have the ankyrin start to spontaneously cluster these sodium channels in the neuroma itself, leading to the spontaneity of pain, which is amazing. And then lastly, this is some more work that I did closely with Sun Zi. We did phosphoproteomics to see, well, is there any phosphorylation changes that occurs in these ankyrin proteins, or any protein for that matter? And we actually found that the ankyrin 2 was, so this is after I did the staining, but we found that ankyrin 2 was actually being uniquely phosphorylated at a specific site at a higher rate in neuromas that were extremely painful compared to control animals, suggesting that this ankyrin isoform is a necessary ankyrin isoform that drives NaV1.8 to the membrane and is responsible for protecting it from endocytosis. And one way to test that is by inhibiting a kinase that is responsible for phosphorylating the sequence. So this sequence corresponds to a phosphorylation by a casein kinase. And I use this casein kinase inhibitor, TBB. And what's really cool and what really had me excited is on the top, I have neurons that were, again, given the scrambled peptide, the NaV1.8 peptide, or the TBB. And we can see there's no difference between the three. However, again, on the bottom, if I give 8-bromocyclic AMP, the cells that receive the NaV1.8 peptide and receive the TBB are exactly the same, suggesting that this mechanism of casein kinase phosphorylation of ankyrin is what's actually driving the protection of NaV1.8, thus increasing the total sodium current and leading to hyperexcitability. So here is just a schematic of the mechanism. Normally, you have the NaV channels in the membrane, um, and NaV1.8 is specifically protected by ankyrin. So I think it's ankyrin B, but we need to do a few more confirmatory experiments to double check that. But once you activate PKA with 8-bromocyclic AMP, or in the case of the naturalistic case, you have CAMP activity, you'll have an endocytosis and removal of potassium channels, increased number of sodium channels, and that drives hyperexcitability. However, when we have the peptide on board, the peptide is able to dissociate ankyrin from NaV1.8, and then NaV1.8 is taken up alongside those potassium channels, thus doing a net washout and preventing hyperexcitability. These are just the conclusions typed out. And looking forward, I want to see if the other NaV channels have a similar relationship with the ankyins, and what other proteins are found bound to NaV channels. It's not just ankyrin; it's a bunch of them. There's more than one. And the world of neuroscience in this project, I think, can bear a lot of fruit. So this is a lot of work. There have been a lot of people who have been helpful in the lab. I made the move from Buffalo, New York to Boston. It's a big change. Buffalo's small, Boston's technically small too, but it's a lot bigger than Buffalo. Also being, a Bills fan and coming to Patriots territory is a little dangerous. That's just a joke. But it was a great smooth transition because of everyone in the lab who helped me out with just okay, acclimatizing myself to the area. And Clifford Wolf has been very supportive of this work. At first, he was very skeptical, but then I was able to show independently with other members of the lab that it does indeed work, and he was really excited. So I've been able to present this at Harvard Medical School and present it to you guys. So I'm really happy for you. I'm very grateful that you brought me here to, share my experience in science and also a little bit about myself and my research. So with that, I can take any questions.
Woohoo! Yeah, that was a wonderful talk, and I think you're getting some kudos in the chat about how amazing your work is. So I'm really, I mean, I have a few questions myself, but I'm waiting for I'll we can wait first if other folks on the call have some as well. But
Sure.
well, I guess like kind of while we're killing time, I'll ask my first question. So I mean, you kind of started the talk really interestingly, talking about like kind of comparing mental, emotional, and physical pain. And I know that a lot of your work focuses on neuropathic pain, but I'm not a cellular person, and I'm curious, can you like kind of speculate about um maybe like as we're translating your work into humans, like how you might see some of like the methodological and procedural approaches, they might be changed as far as incorporating humans' feelings and the subjective experience of pain and things like that.
I'm frozen. Uh, am I back? I
You're
froze
back.
for a second. Okay, okay. So translating this into humans, I do think that there is space to translate this into humans. I did some preliminary experiments with some extra human DRGs that we had in the lab, and I was actually able to recapture exactly what I saw with the mouse DRGs in the human DRGs, which was great. The biggest thing I think when we're taking this, so these peptides are given local, so they're given directly where the injury is, but what Ozempic and semaglutide, tirzepatide, these other lipidated peptides have shown us is that you can give these sub-Q with a once a week injection done by the patient, and they should translate very well. So I think that as far as the neurobiological space and treating pain for humans, I think that can also be done with these peptides. I have to do a little bit of optimization, like dosing and things like that, but I do think that that would be a great way to go about it. And there has been some efforts to develop a oral version of those lipidated peptidomimetics, those GLP-1 agonists. So I think if that technology does become the mainstay, something similar could be done for this because using a very similar technology, it's a different target and different mechanism of action, but the exact same technology in the exact same headspace, I would say.
Awesome. Yeah, I mean, that sounds great. And I love that you had the table highlighting other biotech companies who had done like similar work. And that was a really great approach to, call them out directly, very bold, but I love it. So, oh, Asma has a question.
Oh, please.
Hi, everyone. Wonderful talk. This was extraordinary. I have a quick question about what you think might be some of the side effects of this particular mechanism of action. I think that's one of the challenges with pain medications and analgesics. Obviously,
Mhm,
opioids, it's
mhm.
addiction potential, gabapentins, but just drowsiness, etc. What do you hypothesize with this particular type of drug?
So what's really interesting is the so one thing that I will say is that this peptide appears to be highly specific for NaV 1.8. Now,
Mm,
the issue is that the cell will contextualize the nature of this interaction and it's not driven by the molecule itself. So
mm,
wherever the anchorin is binding to and protecting NaV 1.8, that is where I expect this peptide to bind to. It can bind other nav 1.8 can bind all anchorins. So where we have other anchorins like in the heart, let's say, which is very important, or in the central nervous system, which is extremely important, I am not 100% certain that this peptide cannot or would be able to outcompete the endogenous, let's say, nav 1.5 in the heart or nav 1.6 in the central nervous system. What I think is the best or the most important thing about this is that this interaction is only important when you have ongoing kinase activity. So the
Mm,
biggest thing that I think about is that, well, if you have a healthy individual who's in pain, this peptide will get everywhere, but the interaction is only important in those nociceptors that are highly active. So those in other tissues where you don't have that same activity, you wouldn't expect it to work. But what happens if you have someone who has some type of congenital heart issue where you may
mm,
have this kinase activity? I don't know if cardiomyocytes engage in endocytosis at the same level as the nociceptors. So that's
mm,
something I'm considering and I'm worried about. Another thing that I'm worried about is that in calcium imaging at the cell body, this peptide causes an increase in calcium flux without PKA activity, suggesting that there is a obligate or maybe even an opportunistic calcium channel that is
mhm,
occupying some space on the anchoring molecule when NaV 1.8 can't bind to it. But if we give the exact same peptide and activate PKA, it decreases calcium. And then if you give the peptide to just the axon and look at the cell body, it only
mhm,
decreases calcium. There's not that increase. So I'm worried
mhm,
about if you give it to a healthy volunteer, would they
mhm,
complain about more pain if you give them a stimulus? I'm not too sure. Um, Tried it in mice, and mice can't talk. They can only behave. So the
mhm,
behavior wasn't significant, but in humans, they may say, "Oh, I don't know if they're a little more sensitive or things may feel weird," but
mhm.
I'm not too sure. So my biggest concerns are the cardiac effects as well
Yeah,
as the potential changes in tactile sensitivity.
very cool. Thank you so much.
Of course, of course.
We still have a few minutes left, so we probably have time for at least one or two more questions.
Yeah, I got time. It can be about science. It can be about you know life, growing up in Buffalo, uh, going up in the cold, going from cold to cold.
I mean, I can relate because I'm from Georgia and I lived in Boston, so but
So you already know. You already know.
yeah. I guess on a like a more like career journey kind of level. I mean, so for example, a lot of folks mentioned that when you pulled the quotes that pained you, like that really touched them. Um, do you do you find that you also have kind of like similar sentiments or experiences with folks that you are Black or are underrepresented? Because I guess one question I had was, do you feel like some of those quotes specifically related to the pain of being a Black scientist came from folks that were supposed to be allies or folks that were supposed to be like in your corner, you felt like?
Well, I guess for me, I never really felt like people were supposed to be in my corner. Um, it would be nice, you get a little help every now and then when someone can vouch for you. But those who are supportive have always been supportive. It's always just been an uphill battle, almost like the Sisyphusian effort to try and break into these kind of spaces. I mean, if you just look at the lab picture, the story tells itself. Um, that it's actually a reflection of the floor that I'm on, where I walk onto the floor and I see me, or there's me, and then there's one Black research assistant, and then there is the one Black lab manager that I'll see every so often. It's not like it's not a common experience, especially in Boston, right? So it's always walking into a room and being the first one or the only and stuff like that. So it's very difficult. But I've always had a very strong support system. So like the MedStep, which is servicing the inner city youth of Buffalo, had a lot of Black students, which is great. It was run by a Black woman, and C-Step, which was the collegiate version, was run by a Black woman, which was great. So a lot of it was tailored to, and it accepted underrepresented students. So a lot of it was tailored to the needs of those students, not to just make them good enough, but to make them excellent. Because when sadly, when you're in these kind of spaces and you're giving, 150% or 200%, that is only perceived as 100%. My father always told me you got to be twice as good just to be as good as everyone else. just because you're set back so much further. So, you find yourself at time giving 300, 400%. And again, you fall into those traps of burning yourself out, wondering if you're doing good enough. You're already given 200%. Why am I not publishing for nature publications like so and so in lab down the street? So it's really easy to fall into those traps, but you just have to stay true to yourself, stay true to who you are, and don't feel like you can't lean on that support system. Family, friends. Like I said, my dad and my mom, they have no idea what I do, but they'll still ask me, how's it going? Any new updates, stuff like that. And those are invaluable. Those small interactions are invaluable.
I have a question, if we have time
Sure.
for like a quick
Yeah.
one. So if you could go back in time and talk to the person you were, I guess at the very beginning of your undergrad when you were still in Buffalo, what do you think you'd tell that version of yourself? Just as a bit of advice to pass along to the next generation as well.
Uh, Honestly, I would tell myself to not let the insecurities of others affect your success.
Oof.
So I actually never was I was always confident in who I was. And it wasn't until I started talking to other people that I learned about things like imposter syndrome and learning things about like the concept of social comparison and stuff like that. I was just doing what I was doing and I was having fun, I was having a great time. And then that thought hit me. It was like, that little voice was like, but what if, what if you're not good enough? What if? And then it actually started to get louder and louder until it got to a point where I was actually kind of upset with myself that I allowed that thought to infiltrate my thinking. And it didn't become, and it's still with me to this day, but I constantly just look back at what I've accomplished. And then I realized that that small amount or that small voice was uh, done nothing but inhibit or I guess put a cap on my potential that I see for myself. So now, even though I am still like, man, so and so and that postdoc in that other lab just got a K99. I'm like, you know what? Good for them. I'm happy for them. That affects me no way, shape, or form. I'm gonna keep doing what I gotta do. I know my life is different. I know their life is different. Nope, all PhDs aren't created equal. I'll be all right at the end of the day. So I have to keep with that mentality. And that's actually been very helpful for me. It, moving myself along, especially where we find ourselves with, NIH funding and stuff like that. So, just have to not a positive outlook, but just stay true to who you are.
Yeah, that's really beautiful. If I may just add on, I was speaking to I think she's in grade 12, a student earlier today, and I said, don't let someone else's light make you feel like you're in the shadows. Like you can
Mm,
all be
mm,
light. And I feel like that's
exactly.
really well aligned. So thank you for
I
saying
like that. I'm gonna use that. I'm gonna use that.
that. You better cite me.
Oh, don't worry. I will. I will.
All right, thank you.
Of course.
With that, I do think we're at time. I mean, this has been such a wonderful conversation and I think, everyone here I'm sure is very appreciative of the how approachable you are and how, accessible this talk was for folks. So, yeah, I'm very grateful that I was able to listen in. I'm so grateful I was able to moderate and introduce you and thank you to everyone who joined today. If you have additional questions, Dr. Powell, is it okay if we kind of share any questions that come with you after, if you have any responses for folks who maybe joined late?
Of
Um,
course. Of course. Yeah.
yeah, okay.
That's no problem.
If not, we can arrange a different format, but yeah, so just in case, and if you have any Black and Neuro related questions about our upcoming conference, don't forget to register for Black and Neuro Week in 2025. That's you too, Dr. Powell. We expect to see you there either virtually or in person. So,
All right. I'll
um,
sign up.
yep, and the link should be in the chat for folks. So, yeah, thank you again to everyone. And yeah, we have a great rest of your day.